Okay, this is Raymond Walters. He works with me at the Broad Institute. He got his PhD at Notre Dame in psychology, and he's totally brilliant at everything. He's amazing with data. He's good at computational things. He knows everything about statistics. Primarily these days, he's working on big data projects using the UK Biobank and the GWAS of everything and consequences of LD score regression. He really does everything. So if you have any questions at all, Raymond's a good resource there. But he's going to talk to us about LD score regression, which he's also expert in.
Thank you, Patrick, for that overly kind introduction. So this works out well that we've already had a couple instances today where Patrick and Dan and Isu have pointed to kind of the uses of LD score regression. And so we'll now talk through what that method actually is and what it does. So we've divided this scheduling-wise into two parts. So there's one part today, one part tomorrow. They're roughly labeled as the partition heritability section and the genetic correlation section. We'll see if it actually fully tracks with that, but I'll try to at least mostly stick to that. And so before diving into the method, I feel like it's almost useful to give caveats up front, because I'm going to describe a lot of ways that, like, here's a thing where we make a bunch of assumptions and maybe it sort of works anyway, as maybe analogous to the earlier discussions about things like PCA. But so the why is at least partially just that LD score regression has proven in the about 5 years now since it was published to be a useful tool that is both getting broad use in kind of its various forms and also turning into a foundation for other methods. So in some ways, whatever its benefits or flaws may be, it's also a building block that has gone into other things like MTAG, things like genomic SEM. And so even if you aren't interested in using LD score regression itself, there's some benefit to knowing how it works as it plays into those other things. And it's also, even in the places where it's not the most efficient method, so certainly there are probably other better estimators of heritability, especially if you have individual level data. And I think you're hearing more about those next week, especially. That it's a computationally efficient way at getting at kind of a first pass at maybe some of these analyses of things like heritability and genetic correlation, because it works solely from summary statistics. So if you're without those individual level data, or just don't want to worry about loading your 500,000 or 1 million people worth of genotypes in to do some relatedness estimation, you can do the quick first pass with an LD score regression in probably 5 minutes from start to finish on a heritability analysis. And so there's utility there as kind of a building block QC tool as part of your analysis, even if it's maybe not your end goal product. And so I've already touched on, like, there's the model assumption problems, there's the estimator problems. Also, if we think back to what David said, especially in terms of heritability, like, it's maybe worth some thought as we go through this partition heritability stuff, keeping in mind, like, in what cases is Snip heritability an interesting quantity as it relates to twin heritability or all of narrow sense heritability? In what cases is genetic correlation, like, itself interesting versus do we want some sort of downstream product in terms of more causal estimates, or at least arrows on what direction is causing a correlation? And so things to keep in mind as we talk through all the mechanics of this method. All right, so LD score regression originates in kind of a series of 3 papers that all came out in 2015, kind of co-led by Brendan and Hillary here. For today, we're going to start with this first paper, which is just laying out LD score regression as a univariate method, so not partitioning, just a simple estimator of heritability and this intercept estimate that relates to confounding that's already been mentioned today. And then we'll get into more the partitioning and the genetic correlation pieces both today and tomorrow that are kind of the labels on these sections of the lectures. And so the initial motivation that went into the development of LD score regression was in terms of thinking about inflation in GWAS, which in this sense, in terms of a QQ plot, so this is the null expectation of our p-values. If we just assumed we have our million SNPs are 1 million independent p-values at random from a normal distribution versus our observed p-values. And like this inflation as being at least some index of maybe being worried about stratification maybe we've, have that kind of confounding bias from population structure or other issues, and that's driving us to have p-values that we would want to maybe correct, as has been described, by inflating the standard errors based on genomic control or something. But the observation that if these two QQ plots that are side by side from each other are from, one is a simulated setting where there is truly no signal, it's nothing but population stratification. It's specifically a case where we've done a GWAS of controls from Sweden against controls from mostly the UK. So should be no real signal there. Also works if you just simulate stratification. Versus the other one of these is from a fully polygenic model. So there's no stratification at all. We've simulated some real genetic effects that are spread across the genome. And we're seeing the kind of impact of polygenicity, that there's small amounts of true signal in various places of the genome. And so that kind of drags up the overall distribution above being a null distribution. In a genome-wide sense, because there's lots of small effects. And so being able to distinguish between these two things and thinking about what we may be able to get out of genome-wide data was one of the big motivators for this method. And leads us, I should say, the kind of empirical result that drove the kind of first version of LD score was this Manhattan plot that Andrea showed you from the GWAS schizophrenia that corresponded to a QQ plot that looked like this. And so similar to all the efforts that went into EA and being worried about population stratification, there was very strong concern that you see a QQ plot like this, your GWAS of schizophrenia has actually just discovered some form of stratification, and a bunch of people in the wet lab doing biology are going to waste millions of dollars following up on genes that don't matter and just tell you about what part of Europe you're from. So how might we distinguish between those things? So the intuition for LD score goes something like this. Imagine this is a section of our genome, or we have a toy genome that's only 20 base pairs long, whichever way you want to think about it. And so each of these tick marks is a SNP. And we have some LD blocks. So some of these SNPs are correlated with each other for illustration purposes. We'll say they're perfectly correlated. So things in a block, all just completely indistinguishable from each other in the population. They're a haplotype that travels together. And you have these ones that aren't in an LD block. They're lonely SNPs. They have no friends. And so the thought experiment goes something like, if we have, for some trait, and in a polygenic sense, our effects are distributed across the genome more or less at random. It's a very big assumption, but let's stick to the thought experiment. We'll say it's at random, and so we've gone through our SNPs, and we've dropped down some causal effects of where SNPs have some true effect. If you change that allele in like the Mendelian sense, it would have some true causal effect. And they land here on these particular SNPs. But if you looked at association in your GWAS, you would then see that everything in those LD blocks was associated. So you'd see signal in these couple of LD blocks. You'd see nothing for those two lonely SNPs and for that other LD block. And you can think about, you have some other trait, or you do another random draw of effect sizes in a sense of a random effects model. And you lay down your effects. They, in this case, land here. You see association of these pair of LD blocks. And if you think about this process repeating many, many times, either across different traits or across different regions of the genome, then the intuition that falls through is that your likelihood of ending up associated with the trait in terms of showing up in a GWAS is proportional to how big the LD block you are in as a SNP. So the SNPs here in this biggest LD block are going to show up as associated nearly half the time, because nearly half of this region is part of that LD block. Versus these SNPs that aren't correlated with anything else are only going to show up associated in the instances where they are directly affecting the trait, and thus their likelihood of being associated is much smaller. So kind of the summary there is that if we assume this kind of random distribution of effect sizes across the genome, we'd expect that you should see that SNPs that have more LD friends, they're in these bigger LD blocks, they have stronger LD, should tag more signal, and thus should have more association in our GWAS. And the counterpoint to this is that when we think of these LD blocks in terms of polygenic drift and how it would relate to confounding, is a slightly hand-wavier intuition that because what we're seeing in terms of LD blocks is a reflection of how they're currently transmitting in the population, that the blocks that are together are sticking together because it's taking a long time for recombination to kind of break up things that have become correlated in the kind of population genetics history where those mutations have arisen. And thus, in the sense that these are variants that are continuing to travel together and be on similar haplotypes, the likelihood that those variants have drifted in allele frequency relative to another population is more balanced. It's not affected by the size of the haplotype block. It's just a statement about the likelihood of drift, kind of random regardless of the size of the haplotype. And so where that leaves us is that unlike polygenicity, where your likelihood of being associated is dependent upon the size of your LD block, here, in terms of drift, your likelihood of showing a signal in terms of drift is kind of more uniform, regardless of the LD pattern. And so that kind of idea about the distinction and that there's thus information in LD and how it relates to GWAS signal then leads us to walking through a more mathematical model for how we get to this estimation from LD score regression. And so the starting point for this is the model that we've seen a few times already. Our basic additive model. So remember, we have our linear additive effects of all of our genotypes and some set of covariates here. And we're just, for walking through this, we're going to write this in terms of actually summing over the things rather than the vector notation. And I'm going to make two slight considerations here. One is I'm going to put a star on the beta here for a moment to distinguish so we can have, so we've talked so far in terms of this initial equation being something that maybe we're regarding as a causal model. And so maybe there's some slippage between that causal model that incorporates all possible genetic effects in this A, and so is all the SNPs you observed, maybe a bunch of SNPs you didn't observe because they're rare, all the structural variation, all the other things that we've talked about earlier this week in terms of things that maybe are places for missing heritability, all the things that we aren't directly assessing in terms of SNPs. So in the sense that we're going to put a star here, we're going to say that this is the best linear predictor beta on the basis that we've restricted to some universe of SNPs that we're now summing over. And this will be very integral to thinking about this quantity of Snip heritability as it relates to overall heritability. The other kind of convenience thing we're going to do is just say we've normalized here, both in terms of the variance of the phenotype and the variance of the genotypes. We'll get to kind of a wrinkle that ends up being implied by that normalization, but we haven't made here yet. The last thing I'm going to hand wave, wait on that. The last thing we're going to hand wave a little bit in a minute is a distinction between covariates that exist that were part of our GWAS model, where they were part of our GWAS model, so probably our PCs, probably age and sex and those things, versus covariates that matter in the sense that Patrick's talked through in terms of stratification effects, where maybe there's a true covariate there that's correlated with genotype, but we haven't controlled for. So for the purposes of the notation here, we're going to say that the Z's are things we haven't controlled for in the GWAS, and that this normalization to variance 1 on Y is after residualizing everything that is in the model on those covariates that we included in the GWAS. So this is mostly for notational simplicity. As I walk through this, you can not make that notational statement in terms of how you walk through, as long as you're careful about defining where your heritability parameter is. But for the purposes of walking through this derivation once, rather than like 4 times over mixes of what covariates have included and whether there's stratification or not, I'm going to describe it in that sense and try and keep tabs on that as I walk through. So, given that statement about notational things and where we're starting in terms of a model, we can go back to our narrow sense heritability is, again, our variance from this additive term that's based on the genetics relative to the total variance in Y. With the normalization, and kind of change in notation. We're now just talking about the variance of this sum of these betas, that is the genetic component as written in this linear combination of the betas. And so kind of the distinction that happens, and probably should be an approximately, or a new line here that I realize as I stand here, is that, so this is our clean definition over here of the additive narrow sense heritability. But as we jump here to some restricted set of SNPs, so these beta stars that we're talking about and this normalization that we've done, now we're starting to think more in terms of SNP heritability. So in the sense that maybe we've omitted some things in moving from here to here, that omission means this is now smaller than this, and this SNP heritability is smaller than this total narrow sense heritability. Does that make sense, the logic jump we've made there? All right, I'll take silence as meaning you understand. All right, so again, same additive model I just wrote, and we're going to work through some derivation off of that model. So we're talking about a method that works with the GWAS results, and so we're very concerned about what the GWAS betas mean relative to this model. And so remember I said the Z's that we've left here are Z's we didn't include as covariates in our GWAS, and so what's our GWAS betas here are just the covariance between the Y and our single SNP X, no control for those remaining uncontrolled for covariates. And so just by fitting in the terms as far as what Y is, and then we expand out, kind of distribute out the terms here. So we're going to, so we're good so far on this step. This is just standard distribution of the covariance. But we're going to play a little bit of games here in terms of what we can reorder. So we're gonna, for notational purposes, we're gonna take this covariance between the SNP and residual to be now a measurement error term. We're gonna make the same notational thing that Patrick did earlier, that this covariance between SNP and these unmeasured confounders is now this confounding term we're gonna call S sub j. And then relative to an assumption about the betas being random and uncorrelated with each other, and is there anything else here? And I guess uncorrelated with degree. Yeah, you don't need that yet. I'm jumping ahead to other assumptions. But so we can rewrite this in terms of those same effect sizes, but in terms of sum over the correlation between the SNP you're targeting for this GWAS beta and all other SNPs. And so we kind of laid that out here in words here, but so kind of this breakdown that you have your bias piece, you have your correlation to all their SNPs, aka your count of LD friends, and you have the remaining estimation error since we're talking about a beta hat. Cool. So this is our GWAS betas. I just wrote this in a slightly different order because it gets us to the order that the LD score equations are classically written in. And so here's the list of assumptions we're going to make. This is what I was getting ahead of myself on. So the first is a random effects assumption about these betas. So again, these are the conditional betas from a joint fit of all the SNPs you've chosen to include in the model. And we're going to assume that those effects are mean zero and have some variance. And per what I tried to explain in terms of the relationship between SNP heritability and the total narrow sense heritability, as long as we have independence of the betas from each other, when we're looking at, I'll flip back so that we actually have this here. So the kind of core piece here is that we have SNP heritability that's this term. And so that's a bunch of terms that are just essentially this beta squared and the variance of x, and we've normalized all the x's to have variance 1, so it's just a sum of the beta squares. But there's a bunch of cross terms that we would potentially need to deal with. And so the way we get out of those cross terms, and that allows us to essentially say that the variance of these betas is by itself a heritability without thinking about the covariance terms, is by treating these betas as random and saying that the betas are all entirely uncorrelated with each other. And so what this gets us out of, essentially, is that even though there's covariance between the SNPs, because the betas are random, you don't extract them from the expectation, and so the expectation of the product between the two betas gets you a 0 for that covariance. The other potential way to get there is to simply say that all your true causal effects are not in LD. So instead of having to jump through hoops about random effects and how they're correlated with each other, we just say the causal effects are far enough apart that they aren't having too many issues in terms of these added covariance terms you're missing. And there's a more rigorous walkthrough of essentially, so the other way to frame this is that we're relying heavily on a random effects assumption, so you can walk through this derivation in terms of fixed effects assumption, and that's in a wonderful paper from James and others, I think published last year, that I think is optional on your reading list, so you can find it there. That essentially kind of walks through in a little more detail what's happening there, but essentially we're going to make a strong assumption there that gets us out of those cross terms, so all we're worried about is the variance on these betas. The other assumption that's hiding in that statement about the variance on the betas is that I've said that this variance, which is the variance of a beta for a given SNP k, is constant. And so because I'm making that statement along with having a meta statement in the initial model that we've normalized all the genotypes, implicitly that means I'm making a statement about the relationship between allele frequency and effect size. So that essentially the variance from the causal effect here is inversely proportional to allele frequency, such that the total contribution to variance from a variant does not depend on that variance allele frequency. There are some decently logical reasons we might expect this in terms of rarer variants might be allowed to have larger effects, and that variants of larger effect may be actively pushed towards being rarer based on selection. This also goes back to the figure that Andrea showed, that if we think about allele frequency against effect size, then to a large extent we've spent our time studying things in this range on the basis that things are rare and have small effects, have been largely inaccessible for statistical reasons, and that things that are both common and have large effects seem to be very, very rarely existing. Like by power considerations, they're largely ruled out at this point, especially in behavioral phenotypes. And so, at least to the extent that we think maybe a lot of the effects live in this kind of range, maybe it's reasonable to make that statement about this relationship between allele frequency and effect size. We could optionally specify the model and not make this assumption. So we could, for example, jump to the other end of the spectrum and say that the variance on the effect size has nothing to do with allele frequency, and it's balanced per allele in terms per allele of effect sizes are constant of variance. That gets you an alternate derivation where you then have to keep track of the allele frequencies as part of the model, but is something you could do if you feel strongly about going in that direction. So that's kind of the hiding assumption that's there. The other assumptions we made here are largely in terms of the stratification term. So similar to what I just said about the betas, we're similarly reducing from a statement about confounding at an individual variant to an overall variance. So we're specifically not going to say there's anything different between the SNPs that has to do with their kind of degree of confounding. There's some shared term, and in particular, it's important that any variation in that confounding is independent from the variation in the effect sizes. So that's this assumption here that there's no correlation or covariance between that confounding and the effect sizes. So it's missing a star. And of course, everything is independent of the error term. The last piece we need in terms of the derivation is to assume that we have error variance. It has kind of standard OLS properties, where once we throw in the kind of initial statement that we've standardized the phenotype, we're going to have an error term that's directly 1/1 in terms of the variance. Cool. So with all of those assumptions in mind, we can then say that our chi-square statistic from a GWAS, so as was kind of said earlier, this is roughly the square of the t statistic. It's the square of the t, treating it as having enough degrees of freedom that it's a z, and so we're in a chi-square rather than worrying about degrees of freedom for an f. And so this is just our squared GWAS beta over our estimated variance, or standard error for that. And the expectation is that this variance is just going to show up as sample size. So again, this variance is 1/n, so we just throw n out front. And then we have our squared statement about what the GWAS betas was that we worked out before. And so if we take expectation of this chi-square, distribute the expectation through, then what we have is that, so our expected error variance, our expected, the expected error, we end up in the way it's parameterized previously, ends up that we have error of 1. This corresponds to our null expectation on a chi-square distribution. The mean value of that chi-square should be 1. We have the variance, the expected variance that's coming from this confounding term that, in terms of the LD square equations gets called this A, and the size of which scales sample size by distributing the sample size in. And then we have the piece here, which is, again, this statement about where we had before, we have the effect sizes and the LD scores, or not the LD scores yet, but what will become the LD score is this sense of correlation to all other SNPs, and that some of that affects. Because we're in variance terms, and we've already said that this variance is independent from the strength of the correlation, we can pull that out. And then the last piece of magic here then is that this variance is what we pointed out earlier, is now this Snip Heritability over M. And so this leaves us with what is almost the classic LD score equation, where what we're going to do is we're going to swap in a definition of saying that that sum of LD between variance, so this is the sum over all variance for the LD score of the jth SNP is its sum over all other k, all the variance indexed by k, and so it's the sum of squared correlations there. And so just by notational substitution, we now have what is classically given as the basic LD score equation here. So this null expectation, our confounding piece, and our polygenicity piece that scales with this LD score for all the reasons that we laid out at the outset in terms of the intuition for LD score. Cool. And so as proof of concept, we can take the two examples I showed you at the outset and do this operation. And one way to visualize this is that if we that we bin groups of SNPs based on their LD score, the sum of their correlation to their neighbors, and plot that against the mean chi-square of the variance in that bin. If in this top case, which is the population stratification case, we see no relationship there, whereas in the simulation with polygenic effects, we see this nice linear relationship that we expected from our derivation, but can show in a simulation case that you get this linear connection between the degree of LD the SNP has to its neighbors, measured by this LD score, and that mean chi-square. All right, and this works in real data too. So this is in the supplement of the, I think the first LD square paper as one of the things that was there, but there's a nice illustration here from the earlier GWAS years of education that you similarly in that real data get the nice linear relationship as well. And kind of the strength of the relationship in terms of estimating heritability is apparent in thinking about the slope here. So the slope of this line is what we'll get back to in a second in terms of how that acts as an estimator of SNP heritability. But also the tightness of this relationship is reflective of the kind of the strength of that association signal and the strength of that appearing here versus if you carried this out in terms of where the intercept is here, the degree to which there's some degree of elevation that's not correlated with LD. Cool. So that's most of the theory piece of the kind of core of LD square regression. So now we get into more practical considerations. And kind of the first of those is that, so this is our equation. How do we actually turn this into an estimator? Well, we observe our chi-square statistics out of the GWAS, and we're going to plug in some observed value for LD scores, at which point you have an equation that says you have some observed thing here, some observed thing here. You want to estimate this coefficient, and this is effectively an intercept, and so you just have a simple linear regression. And so the way we accomplish that in terms of plugging in LD scores, I think is on the next slide, but the pieces that gives you as output is, so this intercept, which is a combination of this null expectation of 1 and this index of confounding. And so this is kind of exact, given that A is our kind of index of the amount of variation we're seeing that's excess from stratification or the confounding. This is this intercept then becomes exactly that quantity you'd be looking for in terms of potentially if you want a GC type correction, but you don't want to do genomic control that deflates you for a polygenic signal. You just want to deflate for the confounding variance. This is the quantity you're probably looking for. And then the slope is this in terms of this Snip Heritability. With this kind of piece that we have to make some statement about M. And so I kind of glossed over this before, but so M is number of SNPs. And this is a number of SNPs that are in the universe of SNPs that you specified very back at the beginning when you said we're going to have this model that is the sum of effect from some set of SNPs that may not be all SNPs, but is all the SNPs that we're including in terms of our model for estimation. And so for practical purposes, since we're in GWAS, we're probably not talking about rare SNPs, and we're probably focused on the SNPs that we were able to include in our GWAS and that we're able to get good estimates of LD scores for. I think I have a couple more words on LD scores on the next slide. But so the last piece is that gets you an estimate, but this is an estimate where we've done simple linear regression. We'll get to weights on that regression in a minute. But we've done linear regression with a bunch of SNPs, and the SNPs themselves are correlated. So just the standard like OLS standard errors on this regression coefficient wouldn't be correct. So instead we rely on block jackknife standard errors where we take the blocks of the genome large enough to cover kind of the range of LD and then some, and you do jackknife estimation of this regression coefficient across those. Cool. Right, so where are we actually getting these LD scores from? First thing is we need an unbiased estimator of this R square, so we don't just want to take the Pearson correlation and square it because there's an upward bias there due to overfitting, so we use unbiased R square estimator. And kind of the key things that makes this usable with summary statistics is that we can estimate this LD in some reference sample. So if kind of the ideal scenario is if we know everything about the LD within our sample and we can use those LD scores, perhaps that's the best reflection of the LD structure and thus will give us our best fitting model. But to the extent that LD is fairly consistent within, say, continental populations, or especially if you get down to country level populations, if you're working with UK Biobank or something, it's fairly straightforward to say that, well, we don't know the exact LD structure we have within the GWAS sample, but it probably looks an awful lot like 1,000 genomes or HRC, and so can use that as a baseline to estimate these LD scores. That also actually gives you some separation between any confounding that's driving stratification LD in the GWAS sample versus any stratification LD that would exist in your reference sample in terms of keeping these LD scores separate from that confounding, which might be a nice extra, though benefits that, who knows. However, the downside of this is, we're working with LD from, especially if we use the stock 1,000 genome scores from, 500 European individuals. So there's some noise there, and there's some loss from that kind of mixing and matching between a reference sample and what the actual LD looked like in the GWAS sample. And so we are losing information relative to an in-sample estimate of Snip heritability like you'd get from Gremmel or Hazeman-Elston or any number of other estimates that take advantage of that individual level data. So this is the cost we're paying for the simplicity of something that basically says, download some LD scores, plug in your summary stats from your GWAS, run an OLS that takes 30 seconds, and you have a Snip heritability estimate. Cool. And so since we're defining those LD scores in some external reference sample, we've added yet another layer to our statement about what set of SNPs we're saying is our universe of SNPs that we're talking about the variance explained by for our Snip heritability. And so we now have essentially the convolution of what SNPs were observed in our GWAS, what SNPs were observed in our reference panel, and what SNPs we've chosen to sum over in terms of constructing LD scores out of that reference panel. And so, at least in the scores that are currently in use, there is some intentional juggling, mostly as relates to making our statement about what M should be, that we want to have LD scores that reflect correlation of common variants with slightly less common variants. So say your variant that's at a allele frequency of, say, 6% of the population probably has some correlations with things that are at 3% of the population, but maybe the stuff that's the 3% of the population relative to your sample size and the cohorts involved wasn't terribly stable, and so you don't want to include it in your definition of Snip heritability. And so the current defaults in terms of like software for LD score regression use what gets labeled as M550, which is M that restricts to variants that had a MOF between 5 and 50% in terms of making that statement about M, but still models the degree of correlation in terms of getting the slope for things that were down to a low frequency of, I think, 0.5% in practice based on the 1,000 genome scores. And there's because of the kind of connection of the LD scores and the kind of universe of SNPs and assumptions about how to treat M, at least there's some practical considerations that essentially you're extrapolating from the average effect size you see for the SNPs you plugged into the regression to do your LD score regression to the universe of SNPs you used when constructing the LD scores. And so if there's something biased about the set of SNPs you had to plug into the regression, then you're going to extrapolate from that bias to things where that probably doesn't make sense. And so the most obvious case of this is when your GWAS was on a targeted chip that is something like exome chip, where like half the content is specifically targeted at coding variation in genes and is thus probably of higher effect than your like average SNP anywhere in the genome once you account for all the intergenic regions and such. And so that kind of extrapolation from that kind of data doesn't make a ton of sense. And there's also, for stability reasons of this regression, maybe you don't want to include regions that have very, very strong LD and thus will be outliers with respect to thinking about this regression. Most noticeably the MHC region on chromosome 6, which is kind of this densely packed region that has a lot of genes related to immune function, and so is very, very highly regulated and very complicated in structure, has lots of LD in not nearly the kind of clean blocks that we see in the rest of the genome. And so there, and perhaps kind of the LD that exists from this large inversion region on chromosome 8, there might be value to dropping those. In practice, we drop the MHC, but not the inversion.
So Raymond, can I just
Yeah.
some clarifying set of questions? So, just to reiterate what you said, but I'm just making sure I understand it. So M is a set of SNPs with MAF between 5 and 50% in the reference panel.
Correct.
Okay. And then you're saying that we can do this regression on all SNPs down to MAF 0.5 MAF?
So the SNPs we use in the regression, the default filters are to use SNPs that were at MAF of at least 1% in the GWAS sample.
Okay. Is there any problem with the disconnect between the MAFs of the ones that we're using in the regression versus the ones that are in the LD reference panel?
So it's a problem mostly that it just makes our lives more difficult in terms of stating our interpretation of what this parameter is. So we're kind of already in a weird space and wanting to make a statement about variance explained by a subset of SNPs that we know live with LD to other things that we need to model to not like break our statements about the relationship to LD, but we want to make some cleaner statement about what Snip heritability means as far as some set of SNPs. And so this kind of relying on M550 is just an added layer to that, and so makes it more difficult to say that like it's like we're making a statement about variance explained by effects of common SNPs, but we included rare SNPs in making that statement about the effect of common SNPs, but not all the rare SNPs. And so basically just puts a giant asterisk on what we mean by the estimate, but I'm not sure it's a problem in terms of like there is a quantity there, it's just a very difficult quantity to describe, I guess.
You could estimate LD score regression with like just like a random set of SNPs throughout the genome. Like, you know, you could do it with 3,000 SNPs just scattered throughout the genome. What the random effects, like assuming that the betas are random, what that gets you is that even if we don't observe like the actual betas in our GWAS data, we assume that all the other betas are providing evidence for kind of how big those effect sizes tend to be, and then we extrapolate out because we're in this, you know, they're all random IID sorts of things. And so, so I think that, you know, what's important is like how many SNPs do you think, you know, the SNPs that we're interested in that follow this IID assumption, how many of them are being tagged in the GWAS? And I think the hope is that if we only use SNPs that go down to like 5% or 1% like that, like the maximum LD that can exist between a 5% SNP and a 0.5% SNP is very small. So I think that the hope is that the contribution of these very small SNPs to the GWAS coefficients for these common SNPs is also very small, and so we can just ignore them and say that they're not really part of our analysis.
Yeah, and so I think the other reason to like make the statement about M550 to try and keep them out of our thing that we're estimating is that the lower we allow an allele frequency, the stronger the statement is when we made that reliance on equal variance of per standardized genotype. So across like 5 to 50, like there's some variation, but it's not too bad. You get towards 1%, it's starting to get fairly noticeable. You get below 1%, you're making really strong statements that maybe we don't necessarily have the data to back up, and to the extent that people have looked at it, maybe isn't backed up by the data. And so if you can keep yourself in terms of saying we're going to stick to estimating the 5% and above thing, then maybe that leaves you a little more flexibility in getting away with having made the strong statement about the relationship to a low frequency. Cool. Any other questions?
Yeah, I'm just going to ask a more conceptual question. So the idea so far of talking about LD regression is to estimate heritability of the SNPs that we have in our GWAS. And you mentioned that if we have raw data, we can use different approaches with raw. If you do both approaches on the same sample, would you get fairly similar results? Suppose I get the UK biobank again, as an example, and I do the Gramo, and I get some estimate, and I do the GWAS just on the UK biobank, and I do this process, and I try to see the heritability. Would that give me something that's very similar?
So I have some slides on that I think are in just a couple slides. The quick statement is there are things that are broadly concordant, but between the combination of like nuances about the quantity being estimated and the choice of models and just the different data involved, there's certainly variability there, but I'll get to some more quantitative statements that can be made there in just a minute. Cool. So I mentioned this point about weighting the regression. So we can improve the efficiency of this by accounting for that. So we do the bootstrap standard errors to handle the correlation between the SNPs, but we could also account for that directly in how we weight the regression. There's also an observation of heteroskedasticity, so this was somewhat evident on the plots I showed you before, both from the simulation and from the example to the application of the the education GWAS, but it also just pops out of the model that there's an expectation that not only is the mean chi-square higher as you get to higher LD scores, but also the variance of the chi-square is higher as you get to higher LD scores. And this somewhat makes sense, both it works under an infinitesimal model. It makes even more sense under a kind of spike and slab model where, so amongst your high LD score things, some of them are in LD blocks with an effect, but some aren't. And so, yes, your mean goes up, but you still have to have some representation for the fact that some of those blocks are still truly null. And so you're driving up that variance between where the signal is and where the null distribution is. And so kind of the simple solution to that is that we can take the infinitesimal assumption and use that to drive specific weights that look remarkably like what the regression equation is, because it is just straight, essentially a squaring of that regression as far as what the expected variance is. But it's just to note that, so it's both in terms of what you see when you're looking at that plot of the mean chi-square by bins, there is that expectation about the heteroscedasticity, and that's taken into account, at least in a rough assumption about an infinitesimal model, to improve the efficiency of that regression. And yeah, so there's a bit about correlation between SNPs. You do the jackknife thing for the standard errors, but also you downweight SNPs that are correlated. Kind of the thought experiment here is that if you had a block of 100 SNPs that are all perfectly correlated with each other, you don't really want it to have it changing your regression result whether you included one of them or whether you included all 100 of them in the regression. And so to the extent that if you have all 100 of them in the regression, that you really only want to count it as one data point. And so you downweight relative to the strength of the correlation. Cool. So recap of stuff that I think is perhaps highlighting and reiterating just one more time out of the basic univariate model here. So we're making this random effects assumption about these conditional betas, which implicitly meant we made a statement about the relationship with allele frequency. And this is across all of those betas. So even in like a local sense, we're not allowing for like, these SNPs are next to each other in a gene, so maybe they have similar allele frequencies. No, this model says no. We're summarizing confounding into this single parameter that ends up in the intercept, that is the variance of the stratification term. Again, we're not going to model anything about differences in FST or other things across the variables, just one parameter. We're assuming that our error variance is resembling as if we'd stuck to simple OLS regression for our GWAS. This is a noteworthy wrinkle because it kind of assumes that that we're working with effect sizes that live in a standardization space for OLS, which is not the same standardization space that you get out of something like a mixed model. And so to the extent that the mixed model has improved your prediction of the phenotype and thus dropped the residual variance to some degree versus what you might have expected if you had done a standard OLS regression, there's a little bit of inflation that you might expect to see from running this on mixed model results versus compared to the OLS results it's been derived on. How bad that is strongly depends on how you constructed your mixed model and how strong your prediction off of that variance component tended to be. There's a nice component on the pitfalls of linear mixed models from Alkis Price's group a couple years ago that, in addition to lots of nice things about the mixed models, includes statements about what the expectations are on chi-square statistics, kind of depending on different ways you can have constructed the mixed model. So that's just something worth noting in terms of thinking about yet more biases that might affect these Snip heritability results. We've already talked a decent amount how this quantity of Snip heritability depends on the Snip selection in terms of the LD scores, in terms of what we put in the regression, all that kind of stuff. The efficiency, we're doing these weights based on an infinitesimal model, but whether or not that's the most efficient solution has depended upon, depends on the actual genetic architecture. So something else might work better if there is a sparse architecture. And one bit that I haven't pointed out here because it doesn't matter as much to most of the quantitative phenotypes, but is relevant for disease outcomes, is that all of this machinery was based on working on things in the observed scale. And so if you're working And we're worried about if you're doing the dichotomous phenotype and you're imagining some latent liability scale, especially as it might be relevant if you've oversampled cases versus controls for your disease study or whatever else, that affects then the variance in the population and does weird things to variance components, including to heritability. That there's conversion factors that may need to be required there, but are fairly standard worked out across all Snip heritability methods, which is why I haven't emphasized it too much here, but it's something worth noting. Cool. So you asked for statements about how this compares to other things. So first is a statement against narrow sense heritability estimates, consistent with what I stated might be our expectation because we're talking about a narrower set of variants. But it's nicely observed in practice. The Snip heritability estimates are consistently lower than twin estimates. So this is a comparison across psychiatric diseases, looking at twin data from the Swedish Twin Registry and a bunch of other sources against the kind of current GWAS results for psychiatric disorders in the PGC. And so you kind of can see that the degree of difference varies, how much less the Snip heritability is there in green versus the orange bar. But there is definitely a gap there, as we'd probably expect.
Do you know why certain diseases have a large gap and other ones have a smaller gap? Is that understood?
So there's definitely speculation, and so in some instances there's fairly strong hypotheses about it reflecting, in a proportional sense, a larger contribution of rare variants versus the common variants. So that's particular for autism, which is this one here, and somewhat less, though at least hypothesized, for ADHD next to it as well. That perhaps for something like autism and for young neurodevelopmental disorders in general, that because it's more detrimental over the lifespan, because of the large impacts on fecundity, for especially in autism, that maybe those effects get forced to stay at a lower effect size than for maybe later onset disorders. But that's largely speculative, other than noting that there are solid, like, rare variant sequencing results in autism. So there is some signal there, but whether that's the cause of the gap is hard to say. As far as comparison to other methods, especially in terms of Gremel-based methods, there's a lot of comparisons here that, like, a lot of depth of work in this paper, and so from Matt Keller and Luke Evans and a bunch of other great people who, put in the heroic effort to compare, like, what is this, half a dozen flavors of GCTA and 3 different ways of running LD score and all these other things. I think the quick takeaway is that there is definitely this clear distinction in terms of how well is a relative sense, how much the LD score Snip heritability estimate picks up on rare variants versus how much a Gremel type Snip heritability estimate picks up on rare variants. And so you're seeing that here. So LD score for reference are the ones over here, but so relative to the choice of what the causal model is, when the causal variants are more common, you're seeing closer to the simulated heritability out of LD score here than if its causal variants are either random, and thus including lots of rare variants, or where the causal variants are specifically all among rare variants. And thus you see drastically depleted estimates. Also, just the estimates out of LD score are much more noisy than you'd get out of a Gremel-based method, as kind of suggested up front based on lost information from summary stats. But with that said, if you look across traits, the trends as far as how these methods behave is at least qualitatively similar. So they'll pretty much all say that the Snip heritability of height is higher than the Snip heritability of neuroticism, and kind of have a dynamic range amongst those things, that even if the point estimates disagree with each other, the qualitative statements as far as comparisons is similar. Cool. So I told you all that so we could talk about the partitioned heritability, but I want to pause there also, see if there's any other questions about the mechanics of LD score before we get into partitioning it. Yes?
You're emphasizing LD score regression as like an estimator of Snip heritability a lot, but which surprises me because in the original paper you had all these caveats, and I always thought the stratification interpretation, like the intercept was more interesting than the slope, and that was the sort of how people generally view it. You know. Am I wrong?
I would love if that's the case. I appreciate that that's probably more the case in this room than in the vast majority of rooms. And so I think even though the paper was written that way, it quickly turned into a heritability estimator. And so it does definitely continue to see use in terms of use of the intercept, not so much in terms of people choosing to use it as a GC correction like the SSGAC does, but more in terms of people making the statement about, well, relative to the overall mean chi-square, the intercept is low, and thus our degree of confounding is limited, which is perhaps much more supported as a direct use than trying to make the more quantitative statement about what this Snip heritability estimate means. So I don't want to downplay the value of the intercept, and it's certainly useful in that sense. And to the extent that I'm caveating the value as a Snip heritability estimator, it's because people will often present the results in that frame, and there's lots of necessary caveats about that, versus it's a little more straightforward to take the Snip heritability estimate. We'll get into the partitioning a little more about while it is an estimator of confounding, it also is an estimator of basically everything that doesn't correlate with the LD score, and so you see variation depending on the choice of model. And so maybe there's some wrinkles there as far as use for the confounding also. But I'm much more concerned about potential misinterpretations of the heritability estimator, and that's why I'm emphasizing that so much.
The other thing I was wondering was, you emphasize that the LD scores are measured noisily in most realistic applications. So presumably that will give you attenuation of the slope coefficient. Do you have a sense of how important that is quantitatively? Like, do you know?
So the
scores, basically the scores you make available.
Sure, yeah. So to the extent it's been looked at, which it was looked at some in the original paper, and actually if you go back to the bioarchive preprint for LD score, they suggest that there might be a desire to disattenuate the slope and intercept for, basically adjust for that attenuation. They went through the effort and found that the end attenuation was small enough that it wasn't worth the effort of providing code for it, and that was just based when they were using the 1,000 genomes LD scores. We've revisited it in other purposes, and the degree of noise does seem relatively limited in terms of maybe magnitude of effect, but it's how thoroughly and how across all possible scenarios that's been worked on. Who knows? Yeah.
It's not plausible that that's a major factor in these gaps, explaining these gaps you showed us.
Probably not. So there's probably some small slice of it that relates to that, and it certainly depends on just how different your sample LD ends up being. There's probably good opportunities to play with subsections UK Biobank for exactly that kind of question, but not something I've seen fully fleshed out in that sense.
Quantitatively, I think that the numbers that we produced are something like relative to the variation in LD scores, the error in the LD scores is 2% or something, right? So the attenuation just has to be very, very small.
Yeah.
But that has to be sample specific, right? If I did a GWAS incident sample, that's quite different from any of the LD scores that they make available.
Correct me if I'm wrong, so I think that's based on if you take 1,000 genomes and split it in half, basically.
Okay.
Or maybe it's just bootstrapping to the same sample size, but yeah. Dan.
I'm a little confused by David's first question and the kind of interchange. I would have thought that if you have a biased slope coefficient, then that's going to bias your intercept coefficient. And so why are we less worried about the intercept than the slope?
So you're right in the general sense, and so the attenuation features and things about model misspecification definitely matter there. So there's some things where the attenuation issue that maybe the slope itself is wrong, but there's a lot of the things here about interpretability of Snip heritability that are more about we estimate a quantity and want to say what that quantity means that are not straightforward, but doesn't mean that the slope itself in terms of relationship is.
So I thought when you showed that, maybe I misunderstood, but you showed the graph from Matt's paper that seemed to suggest that the LD score slope was highly variable and wasn't quite getting the right thing, and it looked like there was potentially a lot of error in the slope relative to estimators that you were more confident in.
Right. So so that's getting the right thing in terms of a simulation about the heritability, and so you can be wrong about the heritability, but right about the intercept if the problem with the heritability is something about that you're extrapolating to the wrong universe of SNPs, you've used the wrong M. To some extent, it's, at least for some of these scenarios, it's not even so much that the LD score estimate is wrong, is that it's estimating a different quantity than the quantity that is the true quantity for the simulation. I don't know if you want to chime in on whether you'd agree with that characterization of the comparison.
Yeah, I think that's probably right. I mean, that's what Walters says too. But I mean, we did consistently see that the LD score regression estimates were lower by about 20% than the Grimmel estimates, even for common variants. And once you get to rare variants, it just breaks. It breaks
Yeah,
down.
yeah.
See, that's consistent with just you're using the wrong M. If the M that you have is 20% too large, then all of your estimates are going to be 20% too small, right? But it won't affect the intercept because that's just, it's transforming the slope, which is like correct, but you're transforming it into a heritability, the multiplying by constant.
Yeah.
So is this LD score regression can be used to estimate the cohort-specific heritability, and then can compare different heritability across different populations or time?
So across cohorts, yes, maybe if your cohort's large enough. So LD score is somewhat greedy about sample size to get standard errors that are small enough that you'd likely be able to make any useful comparisons. But if the comparison you want to make is whether like UK Biobank heritability is different than 23andMe heritability, then that's probably a statement that you could try and get at with an LD score heritability, subject to a bunch of other wrinkles that I think we'll talk about more once we get to genetic correlation in terms of those comparisons. Cross populations, no, because you're relying on LD scores here that are specific to a population, so there's no sense in which we're looking at correlations that, so to the extent that LD is different in different populations, there's nothing in this model that allows you to account for that. There's another method called popcorn, which is in many ways an extension of LD score regression, genetic correlation, specifically to look at that kind of question. But otherwise, your ability to make that comparison is really, really thorny. And then, so it was cohorts, populations, and what was the other thing? Sorry, what? Time. Yeah, across time is definitely feasible, at least as long as like you don't have a population with changing demographics where the LD becomes a concern, but as long as you're restricting to a kind of consistent population. In that sense, then across time definitely works, and I think there's some work in the EA paper about that specific question as far as like cohort effects over time for EA. So definitely something that can be done. Yeah.
Yeah, this is a good question. I mean, this might be a little naive, but could you also use this as a quality control as to the degree to which you corrected the population stratification? Because essentially, the two plots you showed, I mean, one is an intercept, a pure intercept, and the other is a pure slope. So could this be used as some kind of a sanity check?
Yeah, that definitely gets done. And so both in the single cohort sense, if your cohort's large enough that you can get a useful estimate within cohort, and certainly once you're done with your meta-analysis as a sanity check that like the component things were doing the right things in terms of control. That's one of the better and more defensible uses of LD score is to look at that intercept. And in particular, you can take a ratio that looks at that intercept relative to the overall mean chi-square of the statistics that gets you essentially a 0 to 1 range value of what proportion of the inflation above null of mean chi-square of 1 is at least suggested to be coming from that inflation of the intercept versus can be accounted for by the slope from the LD scores. Is there one more over there? No, we're good. Where are we at on time? I've got a couple other breakpoints I can use, but
Like 5 minutes.
cool. So we'll get through as much as we can as far as laying out the theory of partitioning, and we'll probably talk about actual results and applications of partitioning tomorrow. So partitioning and heritability, the second paper led by Hillary. And then what we'll talk about here incorporates follow-up led by Stephen Gazal in that same group that kind of extended what kind of annotations we look at for partitioning. So we'll talk about the model in that more general framing. And so the motivation for partitioning is, as we spent a bunch of time talking about, we've made this assumption that there's this single variance of effect sizes that applies to all SNPs. And so we can imagine that there are many places where this probably isn't right, and we're interested in the way it's not right. So we might want to say that variants in coding regions are likely to have larger variance of effect sizes than variants in non-coding regions. Maybe there's some set of genes that's been affiliated with expression in some particular tissue that we think of is of interest to our phenotype, and maybe we want to make a statement about effects in those quote-unquote brain genes versus quote-unquote muscle genes or some other, or against all genes or whatever. We might also want to take that allele frequency assumption, and rather than making the strong statement we made about it being standardized variants per genotype, or making some equally strong assumption about how it relates to allele frequency, maybe we just want to carve up allele frequency into various bins and fit those different categories. This is something that similarly shows benefits when you fit that kind of split in, say, a Gremel framework, so it might be something we would be interested in doing here. And so the idea is to take this single variance component and split it up and say that we'll allow some degree of variation in this variance of the effect sizes contingent upon annotations of the different kind of things. And so kind of, to go back to our model for intuition, what we laid out previously was that we end up with an LD score that in our perfect LD model is just a count of the number of things in these perfectly correlated blocks. But we then might start looking at an LD score that reflects the fact that we have DHS sites, which is a type of is a mark of accessibility relative to how tightly packed the chromatin is. So maybe it's more easy to express things in those regions, or they're more active rather than being stored away. Maybe we're interested in coding regions. We've indicated in different color over there. And so we can imagine that instead of having our overall LD score, we have an LD score that is specifically how many DHS SNPs are you in LD with, or how many coding SNPs are you in LD with. And building a model that says your effect size is going to scale with that information rather than just your total LD, because maybe there are more true effect variants that are living in the variants for those annotations. And so LD to those variants is what matters to what you see in the GWAS.
Just to be clear, those annotations aren't mutually exclusive, right?
Correct, yeah. So we'll be fully general about kind of overlap of annotations. Both, their annotations can overlap. There can be SNPs that aren't in any annotation other than some baseline annotation that includes all SNPs. Also, we're going to jump straight to the generalization where this doesn't even need to be a 0-1 membership in an annotation. This can be some quantitative measure of either fractional membership in something or something else that's a continuous measure that will scale with a statement about the variance of the effect sizes. Good clarification. Cool. And so the generalization we're going to make, as just the kind of the intuition was given for, is that instead of the single variance parameter, we're going to say that the variance of these conditional betas is the sum over a variance term that exists for that annotation. So we have dimensions of the annotation D, and then the annotation of the SNP for that annotation. So in the case that we're talking about just binary annotation, so either a SNP is in a category or not in a category, then this is just 0-1, and this is a statement about how much additional variance there is in these effect sizes for SNPs that are in that category, and versus other SNPs that aren't in that category. And we're summing across all categories, and we'll universally assume that there's always a category that is all SNPs that are in the regression. So there's always some term fitting the variance for SNPs. But so what that implies, given that we've made what is still a fairly strong assumption that the way these annotations combine is additively, what that gives us is that our total Snip heritability should be a sum over SNPs of the sum over annotations of this quantity of the variance. for the same kind of reasons that exist before, in terms of making assumptions about random effects that are uncorrelated with each other, so we have no cross terms, and thus our Snip heritability is thus related just to the squared effect sizes, and our squared effect sizes are just the variance term as captured there. The one thing to note here, though, cool, is that this is a statement about the variance as it relates to the annotation of the causal variant. So we're going to get to LD scores that then depend on the annotation of the SNP you're in LD with, not necessarily you. So if you are in LD with a coding SNP, then you may have higher association, but the causal effects are about that there's higher causal effects in those SNPs that are, say, in the coding region, and thus it percolates out in that sense. Cool. So I'm guessing I'm at like 30 seconds. Okay, so the quick version of the derivation, exactly the same framework. So again, our chi-square statistics, we have our expectations about noise, stratification, and our summing over LD to other things. And all that pops in here then is that these effect sizes now exist with this inner sum over annotation. We reverse the order so it's an outer sum. But so the sum over the annotations. And so you have this effect that is that annotation of the target SNP and the effect size, the annotation of the target SNP in the target annotation, and the effect size, these taus, that are the variance component for that annotation, as they live on top of of the actual LD to that SNP. And so by rearrangement, we then have our same intercept term and a sum over terms that is taus and these new LD scores, which are about the sum over this annotation and that LD. And so that kind of naturally sets up that we'll have multiple regression now, where instead of one LD score, we have a collection of LD scores, and each of them will have an associated slope that relates to this tau, and we'll continue to have an intercept. And we'll talk about in the next session on LD score tomorrow about what this actually looks like in terms of results and in practice and all sorts of cool stuff.